So if we look at the use of nanobodies in infections, this has mostly been done in the context of animal models or in the lab. So for example, nanobodies that have been directed against the hemagglutinin of influenza have been given to mice that have been infected with influenza A subtype H5N1. And if the nanobodies were delivered to the animals who were infected, this greatly suppressed the replication of the virus itself and also reduced morbidity and mortality of the animal. Now, the nanobodies that are generated directly against influenza A will work in similar ways as antibodies, so it will coat it and neutralize it, but it also minimizes the immunopathological effects of influenza infections, specifically a lot of lung damage. In addition, in an in vitro model of Clostridium difficile infection, so Clostridium difficile is a gut bacteria that causes a lot of infections in hospitals, nanobodies against the cell receptor binding domain of virulence factors toxin A and toxin B of C. difficile will actually neutralize the cytopathic effects in fibroblasts in vitro. And this is a really critical point because these nanobodies can also theoretically be directly delivered to patients, although that theory needs to be tested. In addition, in E. coli infections of piglets, nanobodies have been given to these piglets, and actually the nanobody delivery has fully prevented the development of diarrhea in these piglets. So you can see that against viruses and bacteria, nanobodies have played a really large role, and hopefully in the future, nanobodies will be used in the clinic to help battle infections in humans. Now, monoclonal antibodies have truly revolutionized cancer therapy. Immunotherapy of cancer has really made a big splash in the immunology world and the oncology world, and really in a lot of biomedical fields. However, antibody delivery in vivo is really hampered by the large size of antibodies. And in addition, nanobodies, due to their size, can truly penetrate tumors much more efficiently. And once antibodies are inside of a tumor that they can get into more readily because of their small size, they're much more stable in that context. So if cancer cells are creating a harsh environment in which antibodies can be broken down, nanobodies have a much higher chance of remaining stable while they're in the tumor. In addition, nanobodies can cross the blood-brain barrier, meaning that nanobodies could potentially be used to treat a variety of brain cancers that previously have not been able to be treated with monoclonal antibodies due to their chunky size. In addition, nanobodies can be readily modified by conjugating them to drugs, radionuclides, photosensitizers, nanoparticles, or other things, and be delivered to a more precise location. Again, the small size of nanobodies allows for the highly targetable delivery of these conjugated proteins to your tumor. So, although monoclonal antibodies have really made cancer immunotherapy a critical aspect of oncology, the fact that antibodies still aren't the best way to treat cancer leaves a really big question mark in the field. However, nanobodies may be the answer to overcome a lot of the hurdles that antibodies have. And actually, capilacimab is the first nanobody-mediated treatment that's been approved by the United States FDA, and it was developed against acquired thrombotic thrombocytopenic purpura, which is a very rare disease that's characterized by excessive blood clotting in small blood vessels. And nanobodies were generated to target the von Willebrand factor, which is really critical in blood coagulation cascade. So essentially, the presence of antibodies against this specific factor will prevent blood clotting. And this has been highly used and shows that clinical applications of nanobodies are really important for the treatment of diseases. Now, in terms of cancers, the nanobodies are not as readily developed as this specific nanobody that's against this rare disease. However, there's a lot of nanobody-based drugs that are being developed, and a lot of them are involved in cancer. So, for example, multiple myeloma here and some solid tumors, but also against a variety of other different disorders, such as rheumatoid arthritis, psoriasis, and inflammatory bowel disease. So essentially, I guess to summarize, is that nanobodies can truly be used in clinical applications that antibodies have been traditionally used in, such as cancer immunotherapy. However, the small size of nanobodies and their very unique ability to detect epitopes that antibodies can't detect make them a really attractive place for future clinical applications. So now that we've talked about nanobodies, I want to shift a little bit into how we can use viruses in a clinical setting. Now, you might be surprised that I'm thinking about viruses in a clinical setting, considering this is an immunology course. But a lot of what we understand about viruses, we've learned about through the immune system. And we can actually use viruses to our advantage based on their highly unique properties that we'll get into But because we know so much about the immune response against viruses, we can make sure that we're using viruses in a safe way for the host. So there's a variety of ways that viruses are being used in the clinical setting, but today I'm going to be talking about how we can use viruses to induce ectopic protein expression with retroviruses. And the example I'll be going through is CAR T cells. And also, viruses have been used in the clinical setting for very targeted cell killing, and this we'll be talking about in the case of oncolytic viruses. So let's start off with CAR T cells. So CAR T cells are a very exciting, again, new field in immuno-oncology. And the way that CAR T cells work, globally, it's to induce an immune response against the specific cancer or tumor that a patient has. So the way that this works is a patient who has cancer comes in, and a doctor acquires T cells from the patient's blood. So again, this is the physician taking the patient's T cells specifically. And this is important because if we put those T cells back into the human body, of the patient's body, it'll highly minimize the chance of off-target activation or immunoinflammation that we don't want. Then CAR T cells are generated such that a gene for CAR, or the chimeric antigen receptor, is put in. Now, this chimeric antigen receptor is a receptor that's highly specific for the cancer cell itself. So for example, here you can see the cancer cell and this CAR domain interacting directly with the cancer cell. So a doctor will isolate some tumor cells, identify unique molecules on the surface of these cancer cells, generate these chimeric antigen receptors against that specific protein, and put them into the patient's T cells. Then this chimeric antigen receptor that recognizes the antigen once it's in the cell, these cells are allowed to grow out ex vivo, put back into the patient, and then hopefully the CAR T cells attack the cancer cell specifically, allowing for a highly robust immune response against the patient. Now, you might be wondering where viruses come into play here, and that's specifically at this step. So if you have heard about retroviruses are single-stranded RNA viruses that will reverse transcribe their genomes from mRNA into genomic DNA, and then integrate their genomic DNA into the genome of the host, such that these CAR genes are delivered to the T cells that were isolated from the patient's blood using a retrovirus, such that the viruses contain the RNA for the CAR receptor, and this receptor gene is then reverse transcribed and integrated into the genome of the patient's T cell. This way, all of these T cells will be expressing high amounts of the CAR, the chimeric antigen receptor, so that they can be highly effective against battling the cancers. Now, this is a very brief way of how viruses can be used very much upstream of the clinical application. But when we talk about oncolytic viruses can be given to patients to directly kill cancer cells. Now, what are oncolytic viruses? So oncolytic viruses are viruses that preferentially infect and kill cancer cells. So this might seem kind of counterintuitive. If we have a patient who has cancer, why would we give them a virus? That seems like an additional infection, an additional stress on the host. However, if we can generate viruses that specifically kill cancer cells and don't harm the rest of the host cells, that actually can be a pretty good way of getting rid of a large population of cells, i.e. the tumor. The other really cool property of these oncolytic viruses is that although you can give your patient a small aliquot, essentially, of virions to infect the tumor, these tumor cells are going to be killed by lysis, meaning that they will lyse and release their contents into the microenvironment that they're in. This will actually release more virions into the tumor space. And this is really important because you can actually deliver a small dose of viruses directly into the tumor. And then as the virus is replicating in the tumor and lysing cells, you're creating a larger pool of a virion population that can then go on to kill more and more tumor cells. In addition, the fact that there is a viral infection will actually trigger additional anti-tumor responses in the host. So for example, a lot of tumors will actually act in an immunosuppressive manner, such that the cancer will evolve to quiet the immune response of the host to prevent the host from eliminating the cancer cells from the body. The presence of a viral infection will trigger an immune response against the virus. However, the inflammatory nature that this immune response will cause will also allow the body to be reactivated and react against the cancer cells as well, because the activating signals from the inflammation caused by the viral response will be much more activating than the repressive signals coming from the cancer cells themselves. Now, if we look at the history of oncolytic viruses, the idea of cancer regression in viruses has been around for a very, very, very long time. There's been records of people saying that individuals who had tumors who then got sick with a viral infection were magically cleared of the tumor, and this has been in the history books for a long time. However, It was only in the 1920s that there was something that was directly tested to look to see whether viruses can aid in cancer elimination. So this happened with vaccinia virus. So vaccinia virus played a really critical role in eradicating smallpox and was also one of the first vaccines. However, in the early 1920s, Levadetti and Nicolou reported that vaccinia virus can inhibit tumor growth in rats and mice. So they gave rats and mice a variety of different cancers. So these were solid tumor cancers. And then they either infected or didn't infect these rats or mice with the vaccinia virus. And what they found is that those rats and mice that were infected with the vaccinia virus cleared the tumors they had much more quickly and much more efficiently than those rats and mice that were not given the virus. So this was the first time that it was actually reported that that there was a connection between cancer regression and viruses. Herpes virus, after the vaccinia virus, was really one of the first viruses to be adapted to attack cancer cells. And it's kind of the most popular player in this game. And actually, you can mutate herpes so that it replicates, it can't replicate in terminally differentiated and non-dividing cells, but can still infect and lyse cancer cells. And we'll go into a few ways of doing this, of making sure that the virus itself can replicate just in highly replicating cells, which is the main characteristic of cancer cells. Although vaccinia virus and herpes simplex virus are two of the big players in terms of oncolytic viruses, this has been done with a variety of different viruses, and many new viruses are entering this field in terms of utilizing them in different types of cancers. Because viruses can infect different types of cells, and so you can modulate what virus you use specifically for the cancer that a patient has. Now, working with oncolytic viruses, although it has a lot of really great positives in terms of reducing cancer burden, which is a goal of oncology, using viruses is still somewhat dangerous because viruses are pathogens. They can infect the host and they can make the host sick. So when utilizing oncolytic viruses, it's really important in engineering oncolytic viruses that specifically target cancer cells but do not harm the other cells of the host. And this can be done in a variety of different ways.